So we're going to move on to the next talk, which is trying to take all this information about Senolytics and senescence and see if we can translate into humans. And I'm going to try to make my talk about 10 minutes, just in the interest of time, and I think that should be actually doable since there's really only one study in Senolytics in human IPF patients. So what we did basically was a multi-center, multidisciplinary collaborative clinical pilot investigation. So working along with Dr. James Kirkland, who really is a pioneer in the senolytic field over at Mayo Clinic, working with some folks over at Wake Forest and here in San Antonio. And no need to cover this since we already covered what is senescence. And I think Corey actually had a picture of basically IPF being kind of a disease of aging and really, I think, a prime disease that we can look at senescence and senolytics. So just a little bit of background, which some of these, the speakers previously have already touched on, was that senescence biomarkers have been shown to accumulate in the lungs of IPF patients, and there is higher expression of key senescence biomarkers and SAS factors associated with increased IPF disease severity. However, cellular senescence has yet to be targeted therapeutically in humans. And so this cartoon basically just highlighting the idea of trying to use senolytics, dasatinib and quercetin. And dasatinib is an FDA-approved treatment, tyrosine kinase inhibitor, used to treat leukemias. And quercetin, as was mentioned, is a natural plant-based flavonoid that has been shown to induce a basically suicide gene to selectively kill these senescent cells with the aim of thereby mitigating the SAS factor release and hopefully then leading to reduction in the age-related disease and aging phenotypes. So preclinical testing of senolytics D&Q, I think, have already been touched on in bleomycin-induced models. And D&Q has also been shown to improve physical function, body composition, pulmonary function, and exercise capacity in the mouse model. So our study hypothesis was that intermittent oral administration of combination DQ is a feasible and acceptable approach for patients with IPF, and that intermittent DQ is safe and well tolerated in patients with IPF. Primary objective as being a pilot study of first-in-human study just to see about feasibility, looking at recruitment, retention, adherence to the intervention, and assess really the safety and tolerability of 9 doses of DQ administered over a very brief 3-week period, kind of with the idea that this was a sort of a hit-and-run approach, that by knocking out these senescent cells, that there would actually be some durability of response that would not necessarily require ongoing oral therapy or interventions on a repeated basis, and hopefully thereby lessening the chance for adverse events. Some exploratory objectives were assessing the markers of cellular senescence, circulating SAS biomarkers. We had hoped, looking at this in skin biopsies, which I'll talk a little bit about, and also in plasma as well. And we also try to correlate some of that data to mobility and physical function parameters, pulmonary function, pulmonary health, and fatigue from patient-related outcomes. So this was a 2-stage study design, open label phase, and then a randomized controlled trial. And so after getting our baseline measures, essentially the patients were dosed with D&Q for day 1 to 3 during the first week, and we had an adherence check on day 4, and then we repeated that on week 2 and then week 3. And then we had follow-up measures taken at day 22 and 30. Key eligibility criteria for this was stable IPF, pretty standard otherwise, kind of inclusions. The key, I think, also was no evidence of pulmonary hypertension, and the reason for this was that dasatinib has been associated with development of pulmonary hypertension in certain cases, and no anticoagulation with the idea that tyrosine kinase inhibitors may increase the risk of bleeding issues. And patients who were on nintedanib would hold their nintedanib while they were taking the D&Q and then restart the nintedanib on those other days of the week. So the enrollment for this initial open label was 17 patients that were assessed for eligibility, 5 at Wake Forest, 12 at here in San Antonio. 3 were screened out at Wake Forest, not meeting study criteria. 14 patients ultimately were allocated and treated with the intervention dasatinib and quercetin, 12 here in San Antonio, 2 in Wake Forest. No patients were discontinued due to use or lost to follow-up. So we had 14 patients that we could follow up with and analyze. And in our randomized control trial, which we've already completed, we had 12 patients here in San Antonio, randomized 1-to-1 to placebo or to the intervention, D&Q, and ultimately all 12 patients were there for follow-up and analysis. And just comparing the demographics, baseline characteristics between the open label and the randomized, again, small numbers, majority were male, age group pretty consistent with IPF patients, majority had mild to moderate disease, and as you can see, most patients were on either nintedanib or pirfenidone. And so the primary outcome really was this feasibility. This was an open label and pilot study along with their very small randomized control trial. And we did show that we had very good retention rates. Every patient was able to stay in the trial and all the way to the follow-up. Adherence, as you can see, was also very good. We did have one patient of mine accidentally take an extra dose. Not sure if he thought that this was something that would help him or not, but he decided to take one extra. And planned assessments, as you can see, was pretty good in terms of making sure that we got all the measurements that we needed. So safety and tolerability in the open label, there was one serious adverse event, pneumonia and pulmonary edema superimposed on IPF following study completion. And in the randomized control study, we did have one death, but it was deemed to be unrelated, and approximately 2 months later that the patient developed sepsis from cholangitis. And in the open label adverse events, there were 2 reported severe headaches and otherwise mild to moderate effects, which we see a lot in our IPF trials. So nothing that really raised any kind of eyebrows. And in our randomized control study right now, we're still analyzing the adverse events and taking a look at that. In our open label study, the physical function parameters, as you can see, with 6-minute walk distance did show a improvement, albeit, again, just keep in mind that this was not the primary outcome, but this exploratory outcome did look favorable. In addition to gait speed was improved, time chair stands, and the SBPB score. However, the skin biopsies that we did to look for senescence markers unfortunately did not, due to technical complications, we were not able to perform an analysis, and there was no statistically significant change in the circulating SAS factors after 3 weeks of intermittent treatment. But we did see some trends observed regarding moderate correlations between the markers of cellular senescence and physical and pulmonary function. And here are some just highlighting the change in 6-minute walk distance on the x-axis, change in IL-6 on the in the left-hand panel, MMP7 change in the middle, and MCP1. And you can see a correlation that was found during the course of the study. However, obviously, we're looking forward to looking at our randomized control results, which are still right now in the process of being analyzed, and hopefully we'll have that information and that data out. So clearly, study limitations. This is a very small sample size, 24 total IPF patients at 2 different centers. And I think one of the things that we felt based on the fact that we did not see a significant difference in the change of circulating SAS factors was that at baseline, it seemed that there were relatively low levels that we could identify. And this is obviously a short follow-up period, so I think long-term outcomes remain unknown. So in conclusion, this is a first-in-human pilot study of Senolytics in IPF. Short-term, intermittently dosed D&Q appeared to be feasible and acceptable approach that was safe and well tolerated. Obviously, then further larger prospective randomized trials are needed to see if this initial evidence of positive effect on mobility, such as walk distance and other markers of cellular senescence and SAS factors, can be seen. Thank you all very much.